see you all, folks. You'll see here that my choice, or the choice my work has led me to for the encoding triplets of the code are entirely different from Marcos and from Eigen and from others before. They all used the RNY or different versions of that. Mine are entirely different. You'll see about it. But this is just to let it be known from the start. I think the main message of my study here is that protein synthesis is directed by couples of amino acid tRNAs. The strong point here is in the word couples. Then translation is not only the entrance of one amino acid tRNA after the other, because it meets another tRNA inside the ribosome, and it's always a couple. And this was the main idea that led me to work with the code. It started about, in 95? It's a long time ago. The difference, another difference from this model and the pre-existent models is that they start with proto-messenger RNA strings upon which translation develops. I could develop what we call self-referential model. It's due to this initiation of the work with couples of tRNAs and the beginning of everything with encoding tRNAs by themselves. That's what we call it self-referential. And we don't start with long strings of RNA. We start only with pre-existent proto-tRNA oligomers. That's what we can expect from prebiotic chemistry. These proto-tRNA oligomers associate by complementarity of what we call the proto-anticodons, which evolved today to be the anticodons. The couple of tRNAs manifest the transferase reaction. Then I again at difference from Savio and from Francisco, because the transferase reaction is, in this model, a property of the couple of tRNAs that are paired together. You don't need a ribosome to have the transferase reaction. And the other aspect is that the product peptides that are made from these oligomers that are proto-tRNAs, they bind back to their producers that are the tRNAs. Then the product peptides bind to the producing tRNAs, and this is the start of the associations between genes and proteins, or between tRNAs and amino acids, tRNAs and proteins. That's the summary of the talk. I'll have only 6 other slides to show. One is to explain the model. Then we are looking how the pairs of encoded triplets are formed. Then I'll show how we would discover this by studying the hydropathy correlation, which is a very old regularity that is observed from the 60s. It's 60 years ago that this hydropathy correlation was known, but they couldn't interpret it at all. It's only with our work with proteins that we could do it. Then how to encode the modules of pairs of anticodons, then the metabolic explanation for it, and then we'll go to explain in detail the first half of the genetic code. When you see the matrix of the code, which is the matrix here, the green part of it is what we are going to explain, which is the first part of the genetic code. We So we see that there are pairs that can be formed in all those anticodons which are in green here, or in the lighter green here, or other pairs from in the red part, but we'll only concentrate on the green part. The green part is the early part of the code. And my work evolved then from the traditional matrix of the genetic code, that everybody knows. This is modified for my purposes here, which I call a thermodynamic matrix, which has the anticodons full of G's and C's in the middle, and no G's and C's at the peripheral parts. This is what I call the thermodynamic matrix. And then the final results that we'll get to is the hairpin model for the matrix. It's not now any square thing, but it's a hairpin. You see there is a shaft here, and then there is a dome on top of it. We can call it also a champignon model, with the hat of the champignon and then the shaft. Next slide is, it's not coming. Okay. Next slide. This is the model in detail. In the origin, we have what I call a singularity, which are two tRNAs paired together. At the end, we have the ribosomes, which is what we want to explain. And in between here, we have the genesis of genes which are derived from tRNAs. Let me explain first the left side, where we have only two tRNAs paired together. They have to be anti-parallel, like they are here. And in this case, the principal dinucleotide of the anticodon, they are staggered, yeah? Only the central base is aligned. The 3' base of one is in the other, in one side, and on the other in the other side. Then there are wobbles in both sides of the pair of anticodon. This is the only way we can do it. The origin, then what we call the self-reference in it is twice noted here. Once is when they complementarily associate with each other. And the other source of self-reference is when the product peptide binds to the producer tRNAs. That's the first RNP that gets into the process of the living. The genes are modeled here as having originated from pre-encoded tRNAs. They are being encoded by the proteins that associate with them, and then this becomes an RNP entity. And this evolved. The evolution is to have poly-tRNA anticodons all together in one chain. Then you end-chain encoded tRNAs. This is one picture of this. You have proto-tRNAs here in the left. They can associate with themselves, making a chain of tRNAs, and a copy of the surface of these encoded anticodons. A copy of these is the first messenger RNA. Then the messenger RNA is a chain of codons, which are copies of chains of anticodons, which are associated tRNAs into a poly-tRNA. This figure there is from our eldest paper in 1983, 1984. That's about almost 40 years ago. Then the change here, the interesting change is that we get from staggered directions of the anticodons, one to the left and the other to the right. And here they are all aligned. It's because they were aligned here and associated in the surfaces of poly-tRNAs. Then a copy of it is the messenger. And here we are again having the same transferase reaction that was there before, and now it happens here. What happens inside the ribosome is that the tRNAs are fixed in adequate positions to be perfectly aligned and stably, so that this transferase reaction that was very slow at the beginning, now it can be fast because everything is aligned and directional. Next slide will be, how do we understand this mode of pairing that we know? That we have to pair the tRNAs of today in the same way as we think
ignore that email that I sent you.
It happens.
Just ignore it. I've just changed it. We've changed. All right.
Anything with me?
I've just sent you an email.
My name? Okay. For pairing the tRNAs, I took here the image of the tip of the anticodon stem loop of the consensus tRNAs. You see that all of the tRNA is kind of self-complementary. You see, this is conserved here and there, this is conserved here and there, conserved here and there. And if you extend it and make the two tRNAs paired together, it's the same thing. Either they are paired here as they are paired here below. And the anticodon is something in the middle that can be self-complementary, like the laterals of it, or not. The difference in this model is that the encoding anticodons were the non-self-complementary. What is shown here are the self-complementary with G and U. This is, U is complementary to G, and this self-complementarity was found not to be good for encoding. I'll show that in the next slide. But anyway, these pairs of tRNAs can be made with any kind of anticodon in the middle. They can pair being only with the self-complementary sides of the loop or sides of the stem. And one demonstration here is taken from one paper of a group in Belgium, that's Dino Moras and followers. That's from PNAS in 1986. This guy, Dino Moras, is one of the beginners of the crystallography of the synthetases and of tRNAs. And he did this work with that group in Belgium. And this shows exactly one pair of tRNAs where the anticodons are self-complementary. You see the tRNAs has the 3' end to this side, 3' end to that side. Then they are aligned and paired as exactly as it is in this space, in this bottom part of the picture. Then they are self-complementary, and in this way they pair together in spite of the central mismatch. This is the aspartate anticodon GUC paired with itself GUC. And that's exactly why this type of self-complementary anticodons are no good for encoding, because they can pair with themselves. You see, the glycine tRNAs also pair with themselves. And this is no good for encoding. You need to encode one amino acid here and one amino acid there. And when you have the same amino acid being paired with each other, this won't lead anywhere to encode the 20 amino acids. Then this is the main conclusion here. And it will be shown why in the next, in this slide. This work was done in collaboration with Savio, which is here, who is here. And what we did was to re-study the correlation of hydropathies of amino acid residues in proteins with the principal dinucleotides of anticodons. What was known before our study was that everybody obtained this kind of correlation between hydropathies of anticodons or codons, which is the reverse of that, and amino acids. But what was done before us was that they used amino acids bought from the chemical companies, from Sigma Aldrich. And those are amino acids that are put in solution, and then they measure the hydropathies of those amino acids in solution and correlate it with the hydropathies of the dinucleotides and get something like this. And they could never find a way of interpreting with deeper analysis. What our data brought us is that we used protein data, amino acid residues in proteins. When we do this with the same principal dinucleotides hydropathies as before, but now with data from amino acids residues in proteins, then we could reinterpret all that hydropathy correlation and noticed that the principal dinucleotides of anticodons are, they go in pairs. The serine GA anticodon pairs with the serine CU, which is the other anticodon of this sextet of serine. The glycine also pairs with proline and makes, and this set of 4 attributions can be separated neatly from the others. The second step here is you can see you have here all the anticodons with the principal dinucleotide with 2 pyrimidines, UU, UC, CU, UU, you see? All this is 2 pyrimidines. All these are 2 purines. And these are pairs of those, AA with UU, AG with UC, same way as this. But the behavior of these in the hydropathy correlation is entirely different. Here, the steepness, it's steep 45 degrees approximately. This is not correlated. And we could separate these from the other sets of anticodons, which are mixed bases, CG or UG or GU, UA, CA. This is mixed purines and pyrimidines. Here is central G and C, here is central U and A. And we could here then separate the whole 23 attributions of the code into these 3 portions. With further work, we could also separate this middle portion of the mixed bases dinucleotides into two other parts. And when we go to look at the matrix of the code, this figure of pairing of anticodons in this way can only be obtained when we take the diagonals in the matrix. They are never in the horizontals of the matrix. The diagonals, you see the green part here. This makes a pair, which is the serine, glycine, and proline. The blue part here is this second part. These are pairs with these, and these brown reddish part is the mixed sector. That's what I call the homogeneous sector, pairs of two purines with two pyrimidines, and the mixed sector is one pyrimidine and purine here, and the opposite there. And this is the work we proceeded going. I had only 6 slides, that's why they are all full of information, but let's go in parts with it. We are working only with anticodons. In anticodons, we have 48, and here we include also the anticodons that correspond to the termination triplets. This means that we are working with the whole elongation set of triplets, including those that were excluded later when the punctuation developed. And the other exclusion here is one thing that has never been explained correctly in the set of anticodons. When you look at the codons, you have 64. When you go to anticodons, you have at most these 48 that I was talking about, because for each of the boxes of the matrix of anticodons, you have 16 boxes of principal dinucleotides. For each of them,
Romeo, you close your microphone. We don't hear you.
okay, I didn't know I had muted it, sorry. Then we have 48 anticodons, and they are divided into halves. One half is the non-self-complementary, where you have purines in both sides or pyrimidines in both sides, and these can only pair with these. The other half of the code is the self-complementary anticodons. You have a pyrimidine and a purine in the same anticodon, a pyrimidine and a purine. And these sets, I've told already in the first slide that they are no good for encoding. Then the encoding, the primary encoding is for these 16 boxes of the pairs. Let me take only the first one to explain in detail. That's GGG, which is proline, GGA, which is serine, pairing with YCC, which is glycine, and YCU, which is the other serine in the code, which is a sextet. And also an arginine went into here later, but that's due to metabolism that we will examine later. We have here 4 what I call modules of pairs. GGG can pair with CCC, with UCC, with CCU, with UCU. They can all make pairs here. The GGA belongs to the same module because it can also make pairs with the same as those. GGA can pair with CCC, with UCC, CCU, UCU. Then this makes a module of 2 boxes with 2 pyrimidines in the principal dinucleotides, 2 other boxes with pyrimidines, and these can all pair together. They will be differentiated by the stability at thermodynamics. I didn't bring the measurements of stability here. I only put it in a simplified mode, which is more qualitative. Here we have 3 GC pairs. There's 3 Gs here, 3 Cs here, or a U here. Then here only 2 GC pairs. But we can pair, encode first the strongly bound triplets. Then all the others remain to be encoded secondarily. And the fit between this set of pairs, and the code goes exactly for the GGA with its serine and YCU, which is the other serine. Then we have a thing that is strange to me, and the physicists are called to explain it to me. The proceeding for the encoding, the procedure for the encoding goes in 3 steps. First, you encode the strongly stable pair, which is this one. Then the synthetase make the other Y go together with that. You don't encode the unstable intermediate, which are these. These are unstable because they are intermediate in delta G, intermediate in free energy. They are intermediate in stability, but they are competing with themselves too much so that they spoil the possibility of these being encoded there. And then what is left is that you encode the weakest. Then we encode the strongest, and then you encode the weakest. This is facilitated exactly by the exclusion of 5' A anticodons, which are also among the weak. Then the weak anticodons are less perturbed in the pairs among themselves due to the 5' A exclusion. Then you encode the strongly stable and the extremely weak, which are stable just because there is less competition among them. Competition is in the intermediate steps, which are no good for encoding. Then I can do the same, exactly the same rationale for all these 4 boxes. Then I have this mechanism for encoding each one of the 16 boxes of the matrix. This was facilitated by the 5' A exclusion. That's the explanation I have for you. Yeah. What?
Sorry, you have 5 minutes.
5 minutes, okay. Then the other part that needs to be explained is development of the degeneracy. When all these 16 boxes are encoded with the first amino acid, it means that the encoding is referred to the principal dinucleotides, accepting all kinds of wobbles of that box. That's the first meaning of that box. When the box gets second meanings, which is reducing the degeneracy of the box, the second meaning is gets the anticodons that are considered by the first encoding, because the first retains the 5' G, which is stronger and more stable, and they concede 5' Y to the second occupiers. Second occupiers will be directed to the self-complementary pairs, which were not encoded at the beginning. And here I get to the end, which is the champignon model or the hairpin model for the first half of the code. The second half is encoded through mechanisms that don't involve any discrimination by the metabolic pathways. Metabolic pathways are exhausted. The central pathways are already exhausted with the first half of the code. I will explain this step by step. This means that full occupation of the first half of the code, which is the homogeneous sector, 2 purines in one side, 2 pyrimidines in the other side, which is the RNP sector. I will not be able to show you why this is the RNP sector, but it is. This sector in the shaft here is the GNP sector. A nearly complete set of central metabolic pathways is necessary also already to encode that. But I'll be concentrated then here on module 1, which is the glycine and serine, and module 2, which is the leucine and aspartate part of the code. Module 1 then is glycine and serine. This is the beginning of the code, the first encodings that the model leads to. And these are exactly the belonging to the group of amino acids that the Tse-Fei Wang model of coevolution between encoding and metabolism. He examined what he could at that time. The model of his is 1967, I think. So no, 1975. And he couldn't examine this because they didn't know metabolism with that deepness until the time. Then module 1 is encoded with these glycine and serine things. There is a variety of pathways that one of those was reminded to us by Sohan. He asked about the reductive acetyl-CoA pathway, which produces directly glycine and serine with utilization of that acetyl-CoA from this step, plus the amination that gets together. Acetate aminated gets glycine. And with the help of the serine hydroxymethyl transferase, which adds another C1 compound to the glycine, you get serine. This is the first and the simplest of all pathways, which is now filled and integrated into the code. Then you see that there is a variety of pathways that guarantees metabolic robustness to the first encodings, which is the reductive acetyl-CoA, the reductive glycine synthase cycle. There is the serine cycle of bacteria, present-day bacteria, which are the methylotropes. All of them utilize folates to transfer C1 units for the synthesis. What happens next is the serine is deaminated to produce pyruvate, and pyruvate will be then carboxylated again to get oxaloacetate, or it will be duplicated to generate the more complex part of metabolism. But let's first stay with pyruvate. Pyruvate is aminated to alanine. Then, strange, pyruvate is a 3-carbon molecule that derives from the amination of serine, which is already a 3-carbon. Then it's just deaminated, and it's aminated again, but differently from serine. Then it goes to pyruvate, aminated to alanine. And alanine, I am proposing that it is a precursor of the leucine that is presently in those positions of the code. And when then pyruvate is carboxylated to oxaloacetate, it generates aspartate. Then the module 2 of the code, then module 1 is in the left side, module 2 is in the right side, module 2 is alanine and aspartate as first occupiers. And they are all synthetase class 2. Then some of these boxes will get new meanings with evolution. The second meanings to enter the code are what we call the ambiguous attributions, which are phenylalanine, that's here, and lysine, that's here below. The phenylalanine and lysine are what we call ambiguous synthetase classes. Phenylalanine is the classic example of an ambiguous encoding because it's the synthetase is structurally and compositionally class 2, but it acylates the tRNA in the class 1 mode. then it's ambiguous in this sense. It's a class 2 enzyme acylating in the class 1 mode. And lysine is also ambiguous differently from that because there are class 1 in some organisms and class 2 in other organisms. These are the only two ambiguous synthetase systems in the whole code. Phenylalanine is already derived from a duplication of pyruvate that makes gluconeogenesis and the pentose phosphate pathway from which phenylalanine is generated. Lysine is generated by transformations from aspartate. And the final entities, amino acids entering this code are derived already from the citrate cycle that gives alpha ketoglutarate and then glutamate, from which enters proline and arginine. These will be located in module 1. Glycine here is substituted by proline, and serine concedes 5'Y to arginine. And here we get then all the finals of this is the last slide, just to show you again why are the ambiguous class of synthetases. All the small amino acids are class 2, which are those that start the first module: serine, glycine, and aspartate. All those that start that modules 1 and 2 are small and are class 2. The large incorporate those two, phenylalanine and lysine, which are the ambiguous. Otherwise, they are all class 1. Then class 2 is always the small and the ambiguous. The medium-sized amino acids don't discriminate the synthetases. And finally, one last notice to put more value on the metabolism to explain the code. The code is a meeting between amino acids and nucleic acids. But when you go to nucleic acids, when you need pyrimidines and purines, in the metabolism to generate the pyrimidine, there is an aspartate as part of its core. And to generate a purine, there is a glycine in its core. That's why I resist the idea of having the RNA wall. I think we need what was already stated here, an RNP word, not RNA word, to begin with. That's all. I think I didn't get too much into the time of others. Thank you, Savio.